Okay, thank you for the introduction, Hwei. So as I mentioned, today I'm going to be discussing some recent work we've been looking at, investigating the effects of multi-D rTMS on functional connectivity and associative memory. Oops. So by way of some background, in 2014, Jane Wang led a study that was published to Science, which investigated the effects of 5 days of individually targeted 20 Hz rTMS to individualized parietal hippocampal circuits. Impressively, after stimulation, the authors reported quite pronounced, long-lasting improvements in associative memory performance that were highly coupled with a selective increase in functional connectivity across the targeted network. Now, given the potential clinical utility of such a paradigm, namely the ability to non-invasively stimulate the hippocampus and augment related forms of memory, we set about assessing the reproducibility and specificity of these effects using a similar multi-D paradigm. So to provide an overview of our experimental design, we utilized a similar crossover design in which participants completed 2 weeks of brain stimulation. So at baseline, subjects were required to complete a baseline MRI scan, which we used to personalize stimulation targets on the basis of resting state connectivity. Following this, subjects completed 4 days of consecutive 20 Hz stimulation following a 2-second on, 28-second off pattern for 20 minutes, so 1600 pulses per session total. Across the 4 days, and following this paradigm, subjects completed a post-MRI in the first week to allow comparisons of resting state connectivity. So specifically, subjects were randomized into one of 2 conditions, targeting either a region of the left lateral parietal cortex functionally integrated within a parietal hippocampal network, akin to the seminal study by Jane Wang, or conversely an active control site where stimulation was given to a region of the pre-SMA functionally integrated within a sensory motor network and functionally connected to a left putamen seed. This was a counterbalanced crossover design, as mentioned, so subjects completed the opposing week after a minimum of 1 week delay in between. We also assessed associative memory performance before and 24 hours following each week of stimulation using the same face-cued word recall task as used in the seminal paper, which I'll get to in a minute. Overall, we collected data from 40 individuals relative to the original paper, which was conducted with an N of 16 and within a similar age range, the mean age of 25.5 years. So we also followed a similar target localization method to the original paradigm. Namely, for our hippocampal parietal stimulation condition, we seeded a region of the hippocampus, extracted the corresponding time course, and looked for an area within the left parietal cortex that was displaying peak functional connectivity. This region was used as our stimulation target, where we used a neuronavigated TMS. Similarly, in our active control condition, whereby the pre-SMA was targeted, we seeded a region of the putamen, looked for a functional peak within pre-SMA, and targeted this region on each subject's structural image. Now, importantly, when we look at the group level, we see that these networks are relatively spatially independent from one another. So here we have our parietal hippocampal network represented in blue with our stimulation target superimposed here, relative to our sensory motor pre-SMA network represented in orange. And you can see here that they are relatively spatially independent from one another, at least on a theoretical level, suggesting that hopefully we're minimizing interference effects and any crossover effects between conditions. Now, moving on to our associative memory findings. As mentioned, we used the same task as the original seminal paper by Wang et al., where subjects were shown a series of faces presented individually in cards for around 3 seconds each. We read a common English word aloud, and they were required to try and memorize the association between the face and the word that was presented at retrieval or recall time point. Now, we were expecting to see the same improvement in associative memory following parietal hippocampal stimulation. However, we did not see any changes, any net changes group level following parietal stimulation or conversely following our active control site. To make this a little easier, I've also represented these data as change scores, so normalized relative to each subject's baseline performance, aligning with the results presented in the original study. And again, compared to the effect sizes reported by Wang et al., we did not observe a similar trend. So we did not observe evidence of improvements in associative memory following parietal hippocampal multi-day rTMS. So these results are quite interesting because, as I mentioned, we did conduct this study with a larger sample size. So it's calling into question the ability to reliably reproduce these effects with these multi-day paradigms. Now, moving on to our resting state results. So here, we first investigated whether we're able to see evidence of increases in functional connectivity within the hippocampus. So seeding our hippocampus and looking to see evidence of selective increases across our targeted network, aligning with the seminal study, which showed there's evidence of increases from the stimulation target down to the level of the hippocampus. Again, in keeping with our associative memory findings, we did not observe these patterns of changes. We did observe some evidence that there were alterations to functional connectivity between our stimulation conditions, so there were some clusters surviving correction. However, when we interrogate these data and look at the raw beta values, paradoxically, we actually see evidence of a decrease in connectivity following 20 Hertz parietal hippocampal stimulation. And conversely, evidence of an increase following our active control stimulation targeting pre-SMA. So the opposite pattern, really. And again, these regions were outside of our a priori network of interest. So whilst we're unable to reproduce these effects focusing on hippocampal connectivity and seeding the hippocampus, we were next interested in assessing changes in connectivity at the cortical level. So we seeded a region in the parietal cortex corresponding to the stimulation target. And again, we did see some evidence of alterations in connectivity following stimulation. So within plausible regions, within medial temporal regions. However, mirroring the results we observed when seeding the hippocampus, we again observed evidence of decreased connectivity following parietal hippocampal stimulation. And lastly, we're interested in assessing an individual level, in the absence of group level effects, whether there was some evidence that changes to fMRI connectivity were correlated with those observed in associative memory. So again, the seminal study showed quite a strong positive correlation between the two, which, while correlational, provides some evidence that these changes to connectivity might underlie these improvements in memory performance. And again, consistent theme, we were unable to reproduce this result. So some evidence of positive correlation between these measures in our data, but this was not a significant positive relationship. So in summary, increased associative memory and network connectivity have been reported following multi-day rTMS, and we assessed the reproducibility and specificity of these effects. Parietal hippocampal stimulation did not increase associative memory or network connectivity, questioning the notion that we are able to reliably elicit these selective changes in connectivity to augment related forms of learning and memory. Instead, we report some evidence of site-specific changes in connectivity, paradoxically with reduced connectivity across our targeted networks and increased connectivity across distinct non-targeted networks. And overall, these findings suggest a complex interplay between multi-day rTMS and network connectivity. And lastly, just a thank you to all of those who have been involved in this study and the various platforms and institutions that have supported this work as well. Thank you very much.
Thank you. Thanks, Joshua. So excellent presentation. So there's one question from Kate. So can you see the question? So she says, were there any marked differences in participant demographics between the study you were replicating and your own? In particular, with respect to things like age, level of education, sex, and etc.
Thanks for the question, Kate. We compared participants' age and also biological sex, and there were no significant differences on those levels. We also used an algorithm to partition out variance and and try and like a pseudo-randomize and control for other extraneous variables, such as the time of day of stimulation, level of physical activity and education. So we did our best to try and standardize some of those factors.